Greeting to everyone. I am Dr. Asma HLAILI, professor at University of Carthage, Faculty of Science of Bizet, Tunisia. I am director of Laboratory of Phytoplanktonology and co-director of International Laboratory LME COSISMED. I will talk about our paper entitled Efficiency of benthic diatom-associated bacteria in the removal of benzo and fluoranthene. Marine pollution through chemical contaminants has become a matter of great concern because of its deleterious impacts on marine ecosystems. Polycyclic aromatic hydrocarbons are among the most hazardous pollutants with potential harmful effects on biota and ecosystems. They are hydrophobic and ubiquitous. 16 PAHs are considered as priority pollutants. PAHs have strong toxicity, persistence, and bioaccumulation. Therefore, a great deal of attention has been paid to their elimination and degradation. Physicochemical and thermal techniques are used, but they are very expensive with adverse effects on environment. However, biological remediation has been recognized as the most efficient method for decontaminating environments polluted by PAHs. Indeed, PAHs are biodegraded by a wide range of microorganisms, including bacteria and microalgae. It has been shown that a microalgae bacteria consortium could be successfully used in the treatment of aromatic pollutants. There is a synergetic relationship between both organisms. Microalgae provide oxygen required by bacteria for aerobic degradation. In turn, bacteria produce carbon dioxide for algal photosynthesis. More recently, it was shown that bacteria associated with microalgae in the phycosphere can play a significant role in hydrocarbon degradation. Most studies have concerned planktonic microalgae and their associated bacteria, but benthic microorganisms such as diatoms thriving in contaminated sediments can acquire physiological abilities to tolerate and metabolize pollutants. Considering all these findings, the objectives of the study were to assess the role of an indigenous benthic diatom and associated bacteria or benthic diatom alone in the removal of two PAHs, benzoaperane and fluoranthene, to evaluate the structures and function diversity of bacteria associated with the diatom, included attached and free-living bacteria, and to carry out a screening of the isolated strains from associated bacteria for their potential use of crude oil for growth. To answer our objectives, sediments were collected in front of cement manufactory in the Bizerte Lagoon in the north of Tunisia, where high levels of PAHs were recorded with fluoranthene and benzoaperane as dominant compounds. Diatom was isolated and cultured and then identified by team as a species of Nitzschia based on the morphometric characteristics. Associated bacteria to diatom were studied. Attached bacteria and free-living bacteria were sampled by different filtration of the Nitzschia sp culture. Diversity of the associated bacteria were analyzed by the 60S RNA amplicon sequencing. Strains of attached bacteria and free-living bacteria were isolated and identified by molecular tools. The isolated strains were screening for growth on crude oils. PAHs contamination experiments were performed under non-arsenic and sterile conditions, in light and obscurity. All treatments were incubated for 7 days. During the experiments, several variables were monitored, such as chlorophyll A concentrations, cell density of diatom growth kinetics. and past concentration in dissolved and particulate phases. These concentrations allowed us to firstly calculate the percentage of past dissolved in the medium, past accumulated in the cells, and past degraded. Secondly, we calculate the relative contributions of different processes to the past degradation, such as photodegradation, volatilization, adsorption, biodegradation by total bacteria, biodegradation by diatom. Here are the results. This fig shows temporal evaluation of chlorophyll A and growth kinetics of Nietzsche sp under non-actinic and actinic conditions in control and contaminated treatments. This fig indicates that diatom maintained continuous growth under all past treatments. The table shows relative contribution of different past removal processes. Nietzsche sp was able to accumulate benzoapyrene and fluoranthene with different efficiency between actinic and non-actinic culture. Biodegradations constituted the main process of past elimination. Biodegradation was enhanced in the presence of bacteria, indicating the co-metabolic synergy of microalgae and associated bacteria in past removal. Diatom and bacteria show different capacities in the degradation of benzoapyrene and fluoranthene. This fig shows structure of microbial community in attached bacteria and free-living bacteria. Nietzsche sp harbored bacteria communities with a distinct composition between attached and free-living bacteria. The attached bacteria exhibited higher diversity and abundance relative to free-living bacteria. The free bacteria contained genera with the known ability of past degradation, such as Marivita, Erythrobacter, and Alcooligenes. The results of screening for isolated bacteria strains growing in crude oil shows that two strains of Staphylococcus and Micrococcus isolated from the free-living bacteria community show the capacity to grow in the presence of crude oil. The main conclusion of our study was that the indigenous diatom Nietzsche sp from Bizerte Lagoon north of Tunisia with its associated hydrocarbonoclastic bacteria are able to degrade two toxic paths, and the consortium bacteria-associated bacteria could find an interesting application in environmental biotechnology, enhancing the bioremediation of past contaminated sites or in depollution strategies following oil spills. And thanks.